Okay, we're all set. Thank you.
Okay, good. So yeah, brief introduction about myself. As you can see from the background, I'm originally from, oh, I'm so sorry, my phone is going. It's one of those silver alerts. So sorry about that. But yeah, so about myself, I'm originally from Brazil, from Rio de Janeiro. I went to undergrad in Brazil and got a bachelor's in biomedicine, which is pretty much a very similar degree to the biomedical sciences program here in the US, but it's a very, I would say like diverse, because it really opens a lot of doors in a lot of fields within biomedical sciences, very focused towards research. But I myself, been in research and ended up looking for other options, and the biotechnology industry got my attention. I joined KGI in the summer of 2018 for the B-Suite program, and that got me really interested in the Amgen program, which is the one that I ended up getting. So I got the Amgen degree in 2021. So last year I graduated. I was part of the class that was in the middle of COVID, and we had to adjust to the all virtual setting, which was really interesting. And it was a push on flexibility, right, to learn how to navigate that whole situation in the pandemic, which is part of why I chose to have as the theme of my presentation today, the COVID pandemic. And then after I graduated, I joined Amgen. During school, one of my internship was with Amgen, so I was in process development. In the data engineering group, because, as you know, because of the COVID pandemic, everything had to change in 2020. So the internship that was supposed to be in person in the lab ended up being remote. Today, I work at Amgen. So after I graduated, I joined the company in the same department, which is the Attribute Sciences team. But now I'm in a different group. And I deal mostly with like supporting commercial programs. So anything testing related, I interact a lot with QC. So it's a very interesting part of process development, which I'll touch a lot about on today too on the presentation. So moving into the presentation, so our theme today is bioprocessing, the star in the backstage of the COVID pandemic. I guess this is something a lot of people don't talk about, which is how important bioprocessing was in enabling the vaccines and having us get some level of normality after this whole pandemic. So I guess getting started, just talking about the COVID pandemic itself, I guess we all know a lot about the toll, but just giving a couple numbers, we had like over 500 million people infected with the disease around the world, which is a very, very large number. And obviously, sadly, we had over 6 million deaths around the world as well. The US holding, I think we are close to the mark of the 1 million deaths in the US, or we already hit it. So it's really sad how this disease in just a couple of years was responsible for the death of so many people. And then obviously it was a very large scale pandemic that affected over 180 countries. So the dissemination of this disease happened really quickly and impacted all of us. Of course, in terms of timeline, I just wanted to go over how things happened, in terms of timing, all the way from, when the pandemic was declared, all the way until having a vaccine available, right? We know that this timeline is still ongoing because, the pandemic hasn't ended. We are still experiencing, waves of COVID here and there, which will probably continue for a couple of years still until this disease really becomes endemic. But, starting on this, on 2020, January, we had this disease that was kind of being discussed in December of the previous year, this pneumonia that was appearing in China, in this city. And then not really much after, in about a month later, less than a month actually, the World Health Organization declared a global health emergency over this disease because it was just spreading really fast. But then it took a long time for actually restrictions on travel start. So when we got to March, it was already a pandemic. Many countries were having a very large wave of cases. A large number of people got infected. And then, looking at the US, not much after that, we had the declaration of a national emergency. We had the shutdowns and, lockdowns, no one can go out. I guess we all know very much about that. But then a couple months after that, we saw a light, right? After the tunnel, which is having some vaccine candidates that were advancing really fast, that were really promising, using this new technology that a lot of people didn't know much about, which was the mRNA. So we had Pfizer, we had Moderna working, against the clock to get this. We had many operations supported by the government and, worldwide to have vaccines available as soon as possible so we could go back to somewhat normality, right? And then in December, the FDA agreed to have the vaccine. So in less than a year, we went from this is a pandemic and, to, okay, we have a vaccine. And in December 18 of 2020, we had the FDA sign off on Moderna, and then December 11 on Pfizer. So we had those two vaccines in the end of the year that had this new technology, which was the mRNA. So we also know that, to this day, there are many ripples from the COVID-19 pandemic. So we had major impact in inequality and poverty globally. A lot of people had, the kids weren't going to school because we had the pandemic going on. So that impacted like a number of people in a lot of societies because, a lot of people lost their jobs during the pandemic, which was really sad, and it really impacted the world, not only, one country or another, it was everyone on the same boat, right? And then obviously on top of the impact on, economic impact and the burden on education and, poverty, we also had like a massive impact to supply chains and, disruption. So things became more expensive and, we can see the ripples today on like inflation that is like through the roof and all those things across the world. Which is, we have to learn how to deal with it, right? Like okay, until this thing becomes endemic, how do we deal with this? And this has been in discussion across, many leaderships in countries. And then obviously like food systems. So everything's been pretty much messed out, right? Because of this pandemic. And then not only COVID, is an issue, we still have all those other diseases that are still occurring, cancer and, people that suffer with chronic illnesses, they all have been impacted by COVID as well, because of the fear of, going to a hospital, so many cases of COVID, people didn't look for help when they were suffering with like an illness, right? And that's like had a very big impact. We had an increase in number of deaths by cancer this year, the last couple of years because of COVID. Folks, took too long to seek help. And then when they did, they were like a later stage of cancer already. So, in general, we had like a massive effect of this pandemic on our lives, on our economies and everything, right? So one thing that I did want to bring is like how after we had the vaccines rolled out, how this was so impressive, right? How, and this is a quote by Dr. Ezekiel Emanuel. He's like a big guy in public health and epidemiology. And he's, talking about how we have to be super impressed by how the scientific progress that was made in like such a short amount of time was huge, and how that is, so impressive that we were able to get the vaccine rolled out, get the technology, that has been in development for a long time, but most folks didn't know about, that worked out, and that in the market, and then in people's, to the people, right, to the patients. So that was really incredible. And then, a few fun facts of the vaccines, because now we actually have 4 vaccines that are authorized for emergency use or FDA approved. So the Moderna and Pfizer vaccines are actually approved by the FDA. And then we have the Novavax, and there's the J&J vaccine that are also authorized for emergency use. So we have like many options today, right? Not only the mRNA vaccines, but these other 2 types of vaccines that are more traditional. And, there's like a study out there published by the Lancet journal, which is a very well-known scientific journal that researchers estimated how many lives the vaccine saved, and that number is about 14 million people. That is, to me, it's like, wow, these vaccines have been done so much for us, right? It's really incredible. And so many of them have been rolled out. Like we had, and when I saw that number, I was like, oh, wow, that's a very large number. So 12 billion doses have been administered worldwide as of the July 12th. And, of course, we have to consider that 12 billion doses doesn't mean that has 6 billion people immunized. That would be, dream, right? Having almost a very large chunk of the population in the world vaccinated, but we know there's still some resistance. We had the booster shots, and the distribution of the vaccine is not very equal. We have, rich countries retain the majority of the doses, for reasons of, the financial situation as well. But it's still very impressive how we, in like 2 years, had 12 billion vaccines produced and delivered to patients. So that's very impressive. But then also going back to another quote from Dr. Ezekiel Emanuel, like we know there's like so much resistance around the vaccines, around the technology, but just a reminder that the vaccines are one of the greatest triumphs of medicine, right? So they allowed children to live throughout all those illnesses that like would take their lives so early on, diphtheria, polio, hepatitis, and a number of other diseases that we get vaccinated when we're children. And just to emphasize how important this still is to vaccinate your children, not only for COVID, but for any disease, because they are lifesavers. And then going back into that timeline that I showed, like I said, less than 12 months, we had a vaccine rolled out. And I know one of the questions that is in people's heads is like, how did we go from pandemic to having a vaccine in less than a year? And I know that's part of the resistance that people encounter when, deciding to take the vaccine or not. And then I just wanted to go, okay, let's go back. How does this work? How does this process of developing a new drug or a new vaccine, how does that work? So let's think, we have a promising vaccine candidate that went through the very initial stages of, animal studies, which is usually the first step. And it's very promising, looks good. We want to put it in the clinic. There's many questions that would, before this is actually taken to become a product, we have to consider what is our population, right? How many people are going to be the target for this innovative medicine or vaccine? And for COVID, we have to think that we have like 7 billion people in the world that should be immunized because this disease reached so many nations and countries. So the number is very large. And if you think 2 doses per person, that's 14 billion doses, which is a very large number. And then a number of questions pop out, like how do we make sure that we can make this vaccine with quality? How do we manufacture it at a large scale? Because 14 billion is a very big number. How do we optimize the cost of goods, right? We still have to think about cost. How can we optimize keeping the quality? How can we lower the cost so the vaccine can be at the lowest cost possible? How do you manufacture it, ensuring compliance? Because the regulatory agencies are still paying attention to how you manufacture anything. They are and they must be, taking care of that. And how can we improve the stability of the product? That's another very important question in terms of, if you want to deliver this in a very distant place that doesn't have a lot of infrastructure for, keeping, having refrigerators and things like that. So that's a big concern and a big question that comes up. And also, space. How do you optimize the space you have in your manufacturing plants so you can manufacture as much as you can and deliver it, right? So these are a few of the questions that can pop up when you're thinking about, taking a candidate to the next level. And to kind of tie up that, I just wanted to briefly go over the drug development timeline. How does that work? So usually you have like the very early stages, drug discovery, you're looking at the candidates and trying to figure out, okay, what does it work? And, depending on what kind of field you're in, let's say small molecules, which is the traditional pharmaceuticals, you have like over 5,000 candidates at some point. Like you have, high throughput assays trying to figure out what is going to work. And then as you progress through the development stages, you start narrowing down that list of 5,000 to like 200, 200. And then when you get to regulatory approval, you have one, maybe two drugs. So it's a very interesting process, because it's like you go from a very large number of candidates, and then after a number of years, you get to like that one that like, okay. But then there's another part, that's how it connects to bioprocessing. That is the very important one that not many people know about, which is called the process development. So once you discover a candidate that is promising and you want to go to clinical, you have to start developing your manufacturing process, because all those very early stages happen in a very small scale. You're testing in animals, you're not using that material in humans. So all of that actual larger scale manufacturing has to be assessed after you have those candidates that are actually going in humans. So when you start your clinical trials, you are required by the FDA to file what is called an IND, which is a document that allows you to start clinical trials on a molecule. And in that document, you need to include how you manufacture. So a lot of the manufacturing process has to be developed when you start clinical trials. So that process development kind of starts midway as you have like a smaller number of candidates, you start doing that. So when you have your clinical trials start, you have that process somewhat refined, but that process is ongoing. So you have process development happening through the lifetime of your product. So even after it's approved, you still keep that going in terms of like, how can you improve, manufacture more for less, or, improve the technology that you have in your manufacturing process. So it's an ongoing part of the process. And then kind of going into bioprocessing specifically, because I touched on, traditional pharmaceuticals and briefly said something about biopharmaceuticals. So biopharmaceuticals they differ from traditional pharmaceuticals because they require something that is called a bioprocess. So it's not like a traditional chemical synthesis that you mix a bunch of chemicals and then you have your product. It's something more complex because it requires a living cell or living organism or their components to produce that product of interest, which is usually a biological entity like an antibody, a protein, an enzyme, or there's many, the pool of biomolecules is really large, right? And even molecules that don't exist naturally, which is pretty exciting, right? So going into the very basics, so we are familiar with the bioprocess for a very long time. So we have a history of, and records of human made by fermentation from over 7,000 years ago in Eurasia. So as we know, beer, wine, all those goodies, and cheese, they are produced through fermentation, which is a bioprocess. You have living cells, bacteria, fungus, doing a process of converting something into a product. And the case of wine is like, as we know, alcohol, and for beer the same. But then that process is complex, right? But then at some point around 1981, we figured out how to use that bacteria that we had so many processes using those living organisms to actually produce something that is a drug, a life-saving drug that will help people. So going into that, so the first biopharmaceutical that was manufactured is the recombinant insulin. And as you can see here, one of the bacteria that produced it, you're probably wondering, what, E. coli? Isn't E. coli the one that causes, food poisoning or something like that? But yeah, that bacteria was used to produce humulin, which was the first human recombinant insulin available. Because before that, the only option that folks had for insulin, so anyone that was diabetic, they had to extract it from the pancreas of pigs. Oh, excuse me. Sorry, my allergies are super bad. But so basically you would extract the insulin from the pancreases of pigs. And obviously it wasn't human insulin. So people had horrible reactions to it. and then patients with diabetes suffered a lot. So this was actually the first product that really revolutionized the treatment of diabetes, because this is human insulin that was synthesized in bacteria, which is kind of, counterintuitive to a lot of people, but it worked out really well. So the manufacturer of insulin opened the door to this huge opportunity that used recombinant. Probably wondering what is recombinant insulin means. So going back into microbiology and the very basics of it, what is a plasmid? So part of that technology uses bacteria, like I said, and as some of you, anyone that is a little familiar with biology and micro, bacteria, one of the main ways of them transmitting genetic information and obtaining interest genes. I'm so sorry, my throat is super dry. One of the ways that bacteria would transmit their genetic information. So as you know, bacteria can become resistant to an antibiotic, or they can acquire anything that is like beneficial to them. One of the ways that they found to transmit that across themselves, right, because bacteria are very simple cells, was through plasmids, which is basically circular DNA that contains a small group of genes that will allow that organism to produce a particular protein that might be helpful to them. And it's easy. It's a form of DNA that is pretty stable, and it's easily transmittable through the bacteria, and they can do that in multiple ways. So one thing that was discovered, after understanding this process, two scientists realized that wait a minute, we could use those plasmids to make the bacteria produce protein that we want. So Herbert Boyer and Stanley Cohen, they were responsible for doing that. They were both on the on the West Coast. Cohen was in part of Stanford and Boyer from UCSF. And they were able to do that. They basically took a plasmid from bacteria that they knew and understood, and used restriction enzymes, which is basically an enzyme that breaks DNA in specific parts. So in specific sequences of DNA, it digests the DNA and breaks the bond, right? So with that, they were able to use a DNA of interest that had those ends that were very similar to the sequence of the plasmid that was just broken. And then using another enzyme called DNA ligase, they were able to bind that DNA of interest into the plasmid, in making the plasmid have that gene that they wanted to use, which at the time I believe was like GFP, which is a green fluorescent protein, which makes things glow in the dark. And I actually have a fun story that when I was at KGI during B-Suite, I actually did that experiment myself and made bacteria glow in the dark, and that was really exciting. So it's a very simple experiment that you can do in a couple hours, and you have the bacteria the next day expressing your protein of interest. So once that was discovered, obviously a lot of biotech startups started like popping out here and there. So we had Genentech, that was one of the first in the industry, as well as Amgen. So Amgen was one of the first biotech startups at the time to go chase, creating pharmaceuticals from recombinant DNA technologies, which are really exciting. So then after this process, obviously, like you're probably thinking, okay, cool. So you make bacteria or cells, because after a couple of years, we were able to make that work with cells, with mammalian cells. And then that allowed us to like make a very huge pool of proteins. But then that's just the initial point, right? So that's the cell producing what you need to produce. But as you can imagine, if you think of bacteria, you don't want bacteria stuff in your drug, because that can be really bad and cause, reactions in the patients, allergic reactions and fevers, and can even cause death, right? If you take that bacteria liquid and just apply it on someone. So there's a whole process that is needed for that product or that protein to be an actual product that can be used in humans. So starting out with your cell culture, once you narrow down that development process of like making your cells optimally express your protein, then you're going to start your seed train, which is basically you're scaling up your culture so you can create like a very large reactor with it. And then after your seed train is finalized and you have the optimal number of cells to start your reactor, you inoculate your production reactor, which is usually, will depend on the size of you or on your demand. So we have 2,000 liter reactors, but we have, 10,000 liter reactors. So it really varies a lot depending on the target population you might have. But then after that, you have your reactor running, producing your protein of interest for a number of days. It can really vary too on the cell line you're using. Some cells live for longer than others and are stable for longer than others. But then after that, you have to harvest all that liquid and all those cells, all that biomass, right? That chunks of cells that are in the reactor, and you need to separate that from your protein. But you have to do that, thinking that your product is a protein and it's sensible to the environment. So you can't just, do whatever. You have to make sure that whatever you do still keeps your product in good shape. So there are many processes that can be used, usually for bacteria, because they don't produce the protein outside of their cells. You have to do the cell lysis process, which is basically you're smashing the cells. And there's many ways of doing that. So you can get your protein. That process can be really damaging to protein. So obviously, once we moved into the mammalian systems, that process is not necessary because the cells express the protein and they release the protein in this out of the cells. So it's easier to harvest, right? So usually in that case, you would do centrifugation just to separate your solids, which is your cells, from your product, which is in the supernatant, in the liquid. And then sometimes after, you might do like a depth filtration, which is just to make sure you don't have any residue or leftovers of cells that are dead with your product. But then still, that's not enough to make sure you have pure product and not all those other things that could be floating in there, like DNA and proteins from your cell that broke down, which can cause inflammatory reactions in the patients and cause reactions, allergies. So you have the downstream purification process, which will take care of those finer impurities like DNA, like whole cell proteins and all those. So usually for that, chromatography is used as one of the main processes for that. And you have multiple categories of chromatography. You have anion exchange, cation exchange, reverse phase, and all of those will depend on your molecules. So you have to really understand how your molecule structure is and what are the properties that it has, so you can choose your process wisely. So as I go over this, you guys can see how much complexity goes into making a vaccine or making a drug. It's not that simple of like, you have a candidate, it works. You have to also take into consideration all the complexity of manufacturing, right? And then in the end, after you finish your purification process, you have the formulation, which is often overseen, is one of the most critical steps of your product development, because formulations is going to ensure your product is stable for a long period of time across the different conditions. And some proteins and molecules, like the mRNA, for example, they are really sensible to any environmental fluctuations in like pH, salt concentration, all those things. So you need to find a way to protect your molecule so you can have it stable all the way until you deliver to a patient, right? So this is one that in the case of mRNA in particular, it was really critical to be narrowed down because mRNA has been in development for a long time, but that was the one part of the process that they had to work on because they knew all the rest worked out, but that part was challenging. And then obviously at the end, you still have to, fill your vials and figure out how to storage and package and then deliver to your patients. But then, there's so much into biomanufacturing that is like really exciting. So, that was like the traditional kind of way of doing bioprocessing, but there's so much being done today. So we have many technologies doing process intensification. So finding ways of making more of protein and, intensifying so you take less time. Continuous manufacturing. So instead of doing batches of product, which is the traditional way of doing things, or you even fed batch, you have continuous where you have your cells and you keep like switching your media and making sure you're harvesting product at the same time, which is a really nice way of, ensuring you have a larger amount of protein produced at the end of the day, and also making sure your cells are happier because you're keeping them in fresh media for a good amount of time. But continuous is not only restricted to reactors, you can also have like chromatography being continuous. So that's a very interesting technology. There's also single-use technologies who are, I understand a lot of people feel like, oh, but plastics and this and that, but they do save a lot of, there's an environmental benefit of using single-use technologies, which is you avoid the use of harsh chemicals, because the cleaning processes for those reactors is really harsh because you need to make sure there's no residue of bacteria or of a different product before you start another one, right? So when you do a single-use process, you save a lot of time from all those cleaning processes, and as well as using those harsh chemicals and all that waste that is biohazardous. So single-use technologies are really cool and really add a lot of flexibility to the manufacturing process. And recently we've been having so much coming up in the cell therapy bioprocessing, which is like a completely different process for manufacturing, as well as the gene therapies where you're producing viruses. And there's so many things that have to go into consideration to produce a virus, because then your product of interest is something that in a traditional process you're trying to get rid of, right? So it's really interesting. And then even plant-based. So, we are able to make products with bacterial cells and with mammalian cells, but now there's a few companies that are developing plant-based biomanufacturing using plant cells. So they engineer the plant cells to produce a protein of interest. And one of the benefits of plant-based biomanufacturing is that you don't have the presence of a number of viruses that would infect humans like you might have in traditional mammalian cell systems, for example. You don't have those. So like you can save a lot in terms of purification because you don't need to be as thorough as in traditional when you're using bacteria or you're using mammalian cells. So it's really interesting. And not only for biopharmaceuticals, there's many other things. And then finally, we have also what some call the 4.0 biotech, which is data, right? Data has been the hot topic recently, and how to use that to, machine learning, all those technologies to drive your process and improve your process with your historical data and all of those. So there's so much in biomanufacturing that is happening, and it's so exciting. So these is like a overview of all the fun stuff going on. But then going back into the mRNA discussion, right? What does that mean? Like the success of that vaccine, what does it mean? So going back to the basics, what is RNA, right? So RNA is a part of the central dogma of biology, which is, you have DNA, DNA becomes RNA, and then RNA becomes your protein. But then if you look into RNA, its function, if you think about a building, you have your engineer, which would be the DNA, who has all that information on, all the details about that building. And then your engineer will create the blueprints, which we can say that are the RNA. Those blueprints have all the instructions that the workers need to build the building, right? And then if you think of the ribosomes, which are, one of the workers that will produce your protein using the RNA, then you can finally have your building, which is the protein. So that's like an interesting analogy of thinking about RNA as like blueprints to build the building. But then RNA is not only messenger RNA, which is the one that is used to build the building. RNA has many forms. It is a very large family of molecules. So there are many RNAs subtypes, so siRNA and miRNA, which are RNA interferences that actually bar the production of certain proteins. And they are really critical in our body because they ensure that certain proteins that are not needed are degraded before they're synthesized. So these also have been investigated for their potential in therapeutics for cancer and for, a number of other diseases, because they can help remove those proteins that might be helping a cancer or protein that is defective in someone with a rare disease. So there's that group of RNAs that are, they promote degradation or inhibit the production of certain proteins. And also we have the mRNA, which is the one that we are pretty familiar with, that induces the production of proteins, which is really interesting. And it has so much potential, not only in the vaccine space, but in the cell-based therapies, as well as in, as a therapeutic for cancer and other diseases. And then, like I said, it has so much potential, and there's, many, if you go look into the clinical trials.gov or look at biotech companies recently, after the success of the vaccine, we have so many more companies coming into that space of mRNA or RNA therapeutics, or how they call it's genetic therapeutics, because you're using RNA. There's a few developing DNA-based therapies as well. So they can address, from rare diseases, vaccines, as we know, cancer, as well as what they call the undruggable targets. So those diseases that have been really hard to treat because they have targets that are intercellular or, things like that. They're become much more accessible now because you're actually, giving your cell the instructions to make a protein that could be inside your cell already interacting with that one that, might be your target. So it really opens the door to a number of things. And then just to close, thinking about how much progress we've made in the space of, biotechnology, biotherapeutics in such a small amount of time. So if we look here, in about 80 years, we went from having penicillin, which is one of the basics, basic bioproduct, because, as some of you might know, penicillin is also produced by a living organism, in this case, a fungus, Aspergillus. So it is a bioproduct. And today we have mRNA. So we went from having a byproduct that already was produced by an organism to engineering proteins that will be produced by our cells, which is very exciting. And then just to give you a view of how far we came in terms of complexity from penicillin, which is a simple chemical formula, to peptides and proteins. This is the insulin molecule, to monoclonal antibodies, fusion proteins, which don't exist in nature. We just created proteins that didn't exist, to viruses that are engineered to either provide a gene of interest or even kill cancer, to multi-specific antibodies that have multiple, so those things that don't exist, right? Cell therapies, and finally the mRNA. So, this is obviously not the only thing that bioprocessing can do. There's so much beyond biopharmaceuticals. So, specialty bioproducts that can be produced using bioprocess. In that, I mean, enzymes and all those things that are used for things other than pharmaceuticals. So enzymes for industrial processes, diagnostics. There's so many things being developed using phages, bacteriophages. We actually have a professor at KGI that does a lot of very, very exciting work in that space, Michael Corus. And he can definitely tell you a lot about phages because he worked a lot in that space for diagnostics using phages. In agricultural technology as well, using living organisms and molecules that are synthesized using bioproducts to make sure you take care of any pests in your crops, and with that reducing the use of toxic pesticides. And then obviously bioremediation also, there's like a lot of work being done using engineering strains of bacteria to consume. You probably heard of like bacteria that eats plastics and those things. So like there's a lot of work being done in that space too, which is really exciting considering the current, concern with climate change and all those things. So it's a really cool space. And then even cultivated meats, which is another one that is really exciting. So, instead of having animals being grown and, taking so many resources and water and all those things, using biotechnology to produce meat. So that's like a few of the very exciting things going on in this space. And with that, I would just like to close my presentation and open up for questions.
And at this time, you guys are welcome to unmute and ask, or if you're unable to speak, you're welcome to type it into the chat. And I will go ahead and stop the recording at this point, and that way, please feel free to ask away, okay? Thank you.